Thank you for the invitation to present on the development of gene editing therapy for hereditary disease. The human genome contains about 6.1 billion nucleotides, and it is estimated that there is 7,000 different hereditary diseases due to deletion of one or millions of nucleotide, insertion of one or millions of nucleotide, or modification of only one nucleotide. How can we correct these mutations? The initial concept of gene therapy started in about 1980 with Anderson and his colleague. The aim was to deliver a normal gene to compensate for a mutated gene. These concepts apply only to recessive disease. Currently, several gene therapy have been approved by FDA. You can see my review, Mendel et al. In Molecular Therapy, about these already approved treatment. Among these already approved treatment, there is Luxturna and Zolgensma, which use AAV vector. Streamvolins use gamma retrovirus, and there are 6 chimeric antigen receptor T-cell therapy products that have been approved. Dozens of other gene therapy treatments are currently in clinical trial. One of the gene therapy treatments that really impressed me was the treatment of Leber amaurosis.
We can see here he is 2 days before his injection. He's walking with a cane. Here he is 2 days before his injection. He's walking with a cane, holding his parent's hand, walking through the hospital. And similar to the other subjects in our clinical trial, he did his homework using Braille, and then in a classroom was removed from the rest of the children, put in the back of the classroom with a video camera highlighting what the teacher was doing on the board, because they could not see what was going on in the classroom. They could not read books. And this is what Corey can do now. He's now more than 2 years post-injection. He can ride his bike around his neighborhood without anybody with him. He goes to visit his friends. He can play catch. He's a star on the Little League team in his area in New York State. Can play games with small and large.
So as you can see, it's really a fantastic treatment. AAVs were injected in his eye and were able to deliver a gene which was non-functional in his retina. The delivery of a normal gene is not a solution for dominant disease. Thus, several techniques have been investigated to modify a gene, among them meganuclease, zinc finger nuclease, TALENs, and the new CRISPR-Cas9 technology. The problem with the meganuclease and zinc finger and TALEN approach is that each time a gene coding for a gene-modifying protein has to be made, and this takes several months to synthesize this new gene. However, in 2012, the CRISPR-Cas9 technology has revolutionized gene editing because it's always the same Cas9 protein which is used. Only a different single guide RNA is required. This is such a fantastic technology that Dr. Charpentier and Dr. Doudna have received the Nobel Prize for that discovery. The CRISPR-Cas9 technology uses a protein, a Cas9 nuclease, that requires a PAM, a protospacer adjacent motif, which is NJJ for the Cas9 of Streptococcus pyogenes. It also requires the use of a single guide RNA, which has a 18 to 20 nucleotide sequence, which is complementary to the target DNA sequence. This is that part of the single guide RNA that precise exactly where the mutation is going to be made in the genome. In fact, following the complex between the single guide RNA, the DNA, and the Cas9 nuclease, a double-strand break is induced at exactly 3 nucleotides from the PAM. Following a double-strand break in the DNA by the CRISPR-Cas9 technology, as well as by any other technology inducing a double-strand break, there is two repair mechanisms. The first one, and the most frequent, is non-homologous enjoining that results in micro-insertion or micro-deletion of a variable number of nucleotides in the DNA. That approach is frequently used to knock out the expression of a gene because the insertion or the deletion of a number of nucleotides which is not a multiple of 3 nucleotides will change the reading frame, and there will be a stop codon which is encountered after the repair site. This will inactivate the gene, and thus this technique is frequently used to knock out a gene to study what is the function of that gene. The other repair mechanism is homology-directed repair. It requires the presence of a donor DNA which contains sequences which are analog to the sequence that precedes the double-strand break. This sequence is in red here, and a sequence in yellow which is analogous to the sequence which followed the double-strand break. In between the red and the yellow sequence, a blue sequence may be inserted. This can be a few nucleotides, or it can be a complete gene which is inserted exactly at the site where the double-strand break has been produced. However, this homology-directed repair mechanism is not very effective. It is used for genetic modifications of cells in culture, and you need to have a trick to select the genetically modified cell. It is not effective enough to be considered for developing in vivo treatment for any hereditary disease. The CRISPR-Cas9 technology may, however, induce cuts at undesired off-target sites, and this may induce mutation. There are various software which have been developed to predict, using the human genome sequence, where these potential off-target mutation might be located. However, there are sometimes mutations at sites that are not predicted by the software, or the software may predict off-target mutation at site that does not produce off-target mutation. There are, however, methods to identify these potential off-target mutation. There is the guide-seq method and the circle-seq method, and I invite you to read these papers to understand these methods. My laboratory is currently using the CRISPR-Cas9 technology to develop therapies for several hereditary diseases: Duchenne muscular dystrophy, Friedreich ataxia, Alzheimer's disease, ataxia 8, and central core disease. Duchenne and Becker muscular dystrophies are due to mutation in the DMD gene. This is a large gene, 2.4 million base pair, containing 79 exons, and the cDNA itself is 13 kilobase. It codes for a dystrophin protein which contains 3,685 amino acids. Duchenne muscular dystrophy is a mutation of the DMD gene that leads to a severe phenotype. Usually the dystrophin protein coded by that gene is not produced at all, and the patient are bound to a wheelchair between the age of 10 to 12. Becker muscular dystrophy corresponds with a variable phenotype. The patient are bound to a wheelchair between the age of 11, and some can walk until the age of 65. So the phenotype is very variable. It depends what is the mutation exactly in the dystrophin gene, but in every case, some more or less functional dystrophin protein is produced. This is a scheme of the DMD gene containing 79 exons. When an exon, like exon 9, starts with a vertical bar, that means that the first codon in that exon is complete. When the exon ends with a vertical bar, it means that the last codon in that exon is also complete. However, when an exon is terminating or starting with an arrowhead to the right or an arrowhead to the left, it means that the codon is not complete and that one or two nucleotides are needed to complete that codon, and they are present in the following exon. About 70% of Duchenne muscular dystrophy cases are due to deletion of one or several exons, leading to a frame shift. That means that the total number of coding nucleotides which are deleted is not a multiple of 3 nucleotides. And therefore, following the exon deletion, the amino acid will be changed and a stop codon will be encountered. Therefore, the complete protein is not produced. The current treatment for this form of hereditary disease is to induce an additional exon deletion using antisense oligonucleotide to restore the normal reading frame, that is, to produce a deletion, a total deletion, which is such that it is a multiple of 3 nucleotides, and therefore the normal reading frame is restored and the normal amino acids are incorporated in the protein. These antisense oligonucleotide treatments have to be administered weekly, and they cost hundreds of thousands of dollars per year. This is a scheme of the dystrophin protein located under the muscle fiber membrane. Dystrophin forms complex with several other proteins present in the muscle cytoplasm and present in the cell membrane. Please note that the central part of the dystrophin protein contains 24 spectrin-like repeats. Each of these spectrin-like repeats is made of 3 alpha helix, helix A, helix B, and helix C. Please note that helix A is starting on the left side and helix C is ending on the right side, and so normally you have a succession of ABC, 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 and therefore the central part of the dystrophin protein is like a spring. It will stretch and go back to its initial position. The main problem of the exon skipping treatment is that the beginning and the end of the spectrin-like repeat, indicated here by black arrows above the exon number, they do not match with the beginning and the end of exons. Although some Becker patients have a correct reading frame due to deletion of some exon where the total number of coding nucleotide is a multiple of 3, they have an abnormal protein structure. This is the case, for example, of a patient which has a deletion of exon 45 to 47. We have exon 44 directly connected with exon 48, and there is no frame shift. However, the end of helix B coded by exon 44 is fused directly with the end of helix C coded by exon 48, and this gives a kink in the dystrophin protein which is not working optimally, and this produces a severe Becker muscular dystrophy patient. The same is the case also for a Becker patient that has a deletion of exon 45 to 49. Again, no reading frame shift, but the end of helix B fused directly with the end of helix C, and again this produces a dystrophin protein with an abnormal conformation and therefore produce a Becker patient with a severe phenotype. My research group has used the CRISPR-Cas9 technology to develop a different treatment for Duchenne muscular dystrophy. We are using the CRISPR to induce a cut in exon 47 and a cut in exon 58, and therefore the DNA present between these two cuts is removed, and there is a direct fusion between the beginning of exon 47 and the end of exon 58. But the site of these cuts in exon 47 and 58 have been selected such that there is the formation of an hybrid helix B, where the beginning of helix B is coded by exon 47 and the end of that helix B is coded by exon 58. This does produce a normal spectrin-like repeat, and therefore this will produce a dystrophin protein with a normal structure. This approach is a potential treatment for any deletion, any duplication, or any point mutation located between exon 47 and 58. We have tested this approach with myoblasts derived from 3 different Duchenne muscular dystrophy patients. One patient had a deletion of exon 49-50, another one had deletion of exon 51 to 53, and a third one had a deletion of exon 51 to 56. These myoblasts were treated in culture with the CRISPR-Cas9 technology. We have delivered the gene coding for the Cas9 and sequence coding for guide RNAs inducing cuts in exon 47 and in exon 58. And in all 3 cases, we were able to detect the formation of the hybrid exon 47-58 by PCR amplification. We have also done an in vivo experiment using the HDMD mouse model. This mouse model contained the normal human dystrophin gene. We have used an AAV9 to deliver the gene coding for the Staphylococcus aureus Cas9, not the SP Cas9, but the SA Cas9, which is smaller, and 2 single guide RNA. In all cases, we were able to detect the formation of the hybrid exon 47-58 in the heart, in the diaphragm, in the tibialis anterior muscle of that mouse model. We were subsequently able to obtain a different mouse model, the HDMD Delta 52 mouse model, which had a deletion of exon 52, which changed the reading frame, and therefore in this mouse model, the human dystrophin is not expressed. This mouse model was injected IV with an AAV9 coding for SA-Cas9 and the 2 single guide RNA inducing cuts in exon 47 and 58, and we were able to detect the expression of dystrophin in the heart and in several skeletal muscle of this mouse, because this restored the expression of the human dystrophin. Therefore, I am proposing a new treatment for Duchenne muscular dystrophy, which is a systemic delivery of a single AAV coding for SA-Cas9 and 2 single guide RNA to form an hybrid exon, coding for an internally truncated dystrophin protein, which has an adequate spectrin-like repeat structure. And we believe this will be a better treatment than the current exon skipping technique which is used. My research group is also working on Friedreich ataxia, which is a neurological and cardiac disease. It is characterized by coordination problem, decreased reflex, scoliosis, muscular atrophy, cardiac hypertrophy, and this leads to a reduced life expectancy of about 35 to 50 years. Friedreich ataxia is due to a mutation in intron 1 of the frataxin gene. Healthy people have up to 30 GAA repeat in intron 1, and this produces enough messenger RNA for frataxin and a normal level of frataxin. However, Friedreich ataxia patients have up to 1,000 GAA repetition. The longer is this GAA repetition, the fewer messenger RNA for frataxin are produced, and thus the fewer frataxin protein are produced. Frataxin is a mitochondrial protein. In its absence, there is a reduced metabolism of the iron and sulfur, and this leads to oxidative stress, leading to cell death. When the neurons and the cardiomyocytes are dying, they are not replaced by new cells, and therefore there is progressively neurological and cardiac problem developing in this disease. The treatment on which we are currently working is to use the CRISPR-Cas9 technology to delete this GAA repetition in intron 1 of the frataxin gene by using single guide RNA inducing a cut before and after the long GAA repeat. We have initially identified SpCas9 single guide RNA, but now we are using a single guide RNA from a shorter Cas9 called CG-Cas9, Campylobacter jejuni Cas9, to remove this long GAA repeat and hopefully restore the normal level of frataxin expression. Our initial experiments were done with ScCas9 among other things and using 2 single guide RNA, and when a clone was isolated where the trinucleotide repeat had been deleted, there was an increase of 2.54 in the expression of the messenger RNA of frataxin. We have been able to induce the deletion of these GAA repeat in 2 mouse models. The YG8SR mouse model contained about 190 GAA repetition, and the YG8 GR800 contained 800 GAA repetition. In both cases, we were able to induce the deletion of the GAA repeat in intron 1 of the frataxin gene. We are therefore proposing that eventually a treatment of Friedreich ataxia could be the delivery by a single AAV of the CG-Cas9 gene and 2 single guide RNA to delete the GAA repeat. The CRISPR-Cas9 technology is evolving rapidly, and in 2016 there were new modifications of that technology which are called base editing. They permit the modification, the chemical modification of a single nucleotide. In 2016, a cytidine deaminase was fused with the Cas9 nickase, which induced only a single strand break, and with a uracil DNA glycosylase inhibitor. This hybrid enzyme permits the chemical modification of a cytidine into a uracil, which is the equivalent to a thymine, without inducing a double-strand break in the DNA. As mentioned previously, this hybrid enzyme formed by the fusion of a cytidine deaminase with a dead Cas9 or with Cas9 nickase permit the chemical modification of a cytidine into a thymine, and this is done at exactly the position that we want, because this is still using a single guide RNA which recognizes a sequence of 20 nucleotides to position adequately this fusion enzyme. The following year, an adenosine deaminase was fused with a Cas9 nickase. It again permitted the chemical modification of an adenosine into an eosine, which is the equivalent to guanine, without inducing a double-strand break in the DNA. And again, each time, it's the targeted nucleotide which is modified. However, both of these techniques have the problem that if there is several cytidine or several adenosine in a window of 5 to 6 nucleotides, both will be modified. A fantastic new modification of the CRISPR-Cas9 technology was published at the end of 2019. It's a prime editing technology. It uses a Cas9 nickase, but this time it's fused with a reverse transcriptase. And it uses a modified single guide RNA, which is called a PEG RNA for prime editing guide RNA. This uses again the single guide RNA, which recognize a spacer sequence of 20 nucleotides to position exactly in the genome. There is the constant part of the single guide RNA, but this single guide RNA is prolonged by a primer binding site, which is about 15 nucleotides long and will bind with the DNA sequence that has been released by the cut by the nuclease. And this primer binding site is followed by the reverse transcriptase template, which may contain modification of nucleotide, and the reverse transcriptase will use this template to synthesize a new DNA strand that will contain the exact modification that we want. This technique permits to replace any nucleotide by any other nucleotide, and several nucleotides may be simultaneously modified if we want. The pegRNA, which is required to do the specific nucleotide modification, is very easy to assemble. You start with the plasmid, which contains the red fluorescent protein, and this red fluorescent protein gene may be removed by BSA1 digestion. You are then left with the first part, which is the backbone of that plasmid. And then there is the pegRNA spacer sequence, which is the sequence that will permit to target a specific nucleotide sequence. This sequence is simply ordered from IDT. And then the third piece is the pegRNA extension annealing oligo. It contains the primer binding sites and the reverse transcriptase template. And again, this sequence may be simply ordered from IDT. And finally, the fourth part is the constant backbone of the single guide RNA, which will be the same for all the different pegRNA that you want to assemble. So therefore, various pegRNA can be very easily designed. You can design 10 different ones to target the same modification that you want to make. Usually the length of the primer binding site and the length of the reverse transcriptase template are varied, and this changes the efficacy of the pegRNA. But these pegRNA, 10 of them may be ordered in the same day from IDT, and they will come within a few days, and you can do your experiments. As mentioned before, the sequence of nucleotide in the reverse transcriptase template may contain modification that will be introduced in the new DNA strand, which is synthesized. About 30% of the mutation responsible for Duchenne muscular dystrophy are single nucleotide modification. When we initially wanted to use the prime editing technology to correct point mutation in the DMD gene, we did not have access to cells of patients containing such point mutation. So our initial experiment aimed at introducing these point mutation to eventually use the cell to correct the point mutation. So the first step is to identify the PAM for the SPCas9 case, and in this case, the PAM for SPCas9 is NJJ. So we want to modify the CGA codon into a TGA codon, which is a stop codon. And as you can see in this slide, we have ordered 9 different pegRNA, which contain various lengths of primer binding site and various lengths of reverse transcriptase template. When we do such prime editing experiment, after the treatment, we extract the DNA of the cell and we amplify by PCR a sequence of about 300 nucleotides in the region of the gene that we want to correct. And then these amplicons are sent for deep sequencing technology using the Illumina technology, and we are returned by the Genome Center 10,000 amplicon sequence, and we can analyze the sequence by computer and determine how frequently the desired mutation has been made, And we can also verify if there are insertion or other type of mutation that we did not want. When we initially started to use the prime editing technology to introduce point mutation in the DMD gene, we were able to obtain about 6% of the desired point mutation. But as you can see in this slide, other gene like the EMX1 gene that was used initially in the paper of Enzalone, we obtained 32% modification of that gene. One of the modification of the prime editing technology, called PE3, permits to increase the frequency of the desired mutation. The PE3 technique uses, in addition to the PEG RNA, a single guide RNA to induce a single nucleotide strand cut, but in the opposite strand than the cut made by the PEG RNA. And this production of an additional cut at some distance from the cut done by the PEG RNA permits to increase the percentage of gene modification. We have used this PE3 technique to induce modification in the DMD gene, and indeed the insertion of an additional nick at some distance from the PEG-induced nick did improve the success of the genetic modification. Another method to improve the efficacy of the prime editing technology is to change an additional nucleotide to induce a mutation of the PAM, the protospacer adjacent motif, so that the Cas9 can no longer bind to a strand of DNA which has already been modified. Therefore, with this approach, two simultaneous mutations are introduced, one to introduce the stop codon and the other one to mutate the PAM, but this is done by the same PEG RNA. Using these two improvement methods, i.e. inducing a nick at some distance from the nick induced by the PEG RNA by using an additional single guide RNA, and inducing a mutation of the PAM with the PEG RNA introducing this additional mutation, we were able to obtain up to 37% gene modification in exon 35 of the DMD gene after a single treatment of human cells in culture. We have already used the prime editing technology to introduce additional point mutation in the DMD gene in exon 20, 43, 55, and 61. That technology, that prime editing technology, works very well, and we've been able to use it to modify nucleotide in several other genes. A potential treatment for Duchenne muscular dystrophy is that the modification of a few nucleotides using the prime editing technology. This can be used, of course, for DMD due to point mutation, but this may also be used to DMD which are due to exon deletion, because this will permit to restore the normal reading frame without deleting an additional exon. My research group is also working on the development of a preventive treatment for Alzheimer's disease. Alzheimer's disease is due to the abnormal metabolism of the amyloid beta precursor protein, APP, by the beta secretase. In fact, this amyloid precursor protein, transmembrane protein, is normally degraded by a cut by the alpha secretase, followed by a cut by the gamma secretase, and this produces protein fragments which are degraded without causing any problem. However, the amyloid precursor protein may also be cut by the beta secretase, followed by a cut by the gamma secretase, and this produces short beta amyloid peptides which are 40 to 42 amino acid long. These beta-amyloid peptides aggregate to one another. They form the amyloid plaques in the brain, leading to the death of neurons and to the memory problem. This scheme illustrates the amyloid precursor protein. In fact, we have the sequence of the amino acid near the transmembrane domain. All the position of the beta secretase, alpha secretase, and gamma secretase are indicated. All the amino acids with a star above their name are amino acids which are mutated, leading to early onset Alzheimer's disease starting at about 35 to 40 years old. These people have memory problem. Please notice the alanine in position 673 indicated by a red arrow. When this alanine is changed for valine, the person have Alzheimer when they start to be about 45 years old. However, when this alanine is changed for threonine, the person that have this mutation are prevented from being Alzheimer even when they are 95 years old. This is the Icelandic mutation that has been described by Johnson et al., Nature 2012. And this mutation was observed in 0.1% of the population in Iceland, and therefore this is called the Icelandic mutation. It's a mutation that protects against becoming Alzheimer. Therefore, my research group made experiments aiming to introduce this protective Icelandic mutation H673T, to introduce this mutation by the prime editing technology in the amyloid precursor protein gene. And therefore this would be a treatment, a preventive treatment for Alzheimer. Therefore, for these experiments, our hypothesis was that the progression of Alzheimer's disease could be slowed down or even prevented by introducing the A673T mutation. And to introduce that mutation, all we have to do with the prime editing technology is to change one nucleotide in the alanine codon to change it into a threonine codon. To introduce this desired mutation in the amyloid precursor gene, we use an approach where we simultaneously induce the modification of alanine into a threonine, but we also modified one of the nucleotide of the PAM to prevent a subsequent binding of the SP-Cas9 protein. When we initially did these type of experiment, we were able to introduce the desired mutation in about 18% of the cell, but when this treatment was repeated 10 times, we were able to obtain up to 60% of the APP gene that contain the A673 mutation. The prime editing technology, which is already a fantastic technology, was recently further improved by David Liu's group. David Liu's group initially observed that the extension of the single guide RNA containing the primer binding site and the reverse transcriptase template were degraded by exonuclease present in the cell. They are not protected by the SP-Cas9 nicase. To prevent this degradation of the 3' end of the PEG RNA, they have further prolonged the PEG RNA, making it into an engineered PEG RNA that contains a pseudo-knot sequence. In fact, it's the RNA is folding on itself and forming double-strand RNA, which is protected from degradation by the exonuclease. And this modification of the PEG RNA further improved the prime editing technology by 2 to 3-fold. We have recently confirmed this improvement in my laboratory for modification of the DMD gene. The group of David Liu has also describe a second improvement of the prime editing technology. They have modified the Cas9 nickase so that it is not inhibited by mismatch repair protein. They have also added another nucleolocalization signal to improve the delivery of the fusion protein, SPCas9, fused with the reverse transcriptase to improve its delivery to the nucleus. And they have codon optimized this reverse transcriptase. This is another improvement that improved prime editing by 2 to 3-fold. The main problem of gene therapy and also of gene editing technology is the delivery. People are usually using adeno-associated virus, but to deliver the Cas9 nickase fused with a reverse transcriptase, 2 AAV would be required. An alternative delivery method is to use lipid nanoparticle to deliver the messenger RNA and the single guide RNA or the PEG RNA with lipid nanoparticle. This technique has recently been used in an article in Nature, and it was also used by the Verve Therapeutics. When I prepared this conference, I looked back at the discovery of the DNA. It's only in 1953 that Crick and Watson proposed the DNA structure. When I was a student, undergraduate student at McGill University in 1970, people were starting to sequence virus. And just sequencing 100 nucleotides of a virus was a PhD lasting 3 to 4 years. And I thought, never will they sequence the complete human genome before I'm dead. In 1980, the concept of gene therapy was proposed by Anderson et al. In 1990, the Human Genome Project was launched. And I'm not dead, and yet they have already successfully sequenced the complete human genome. In fact, the complete human genome has been sequenced for already thousands of people, and the cost of sequencing the complete human genome is now about $1,000 per person. In 2012, the correction of mutation by the CRISPR-Cas9 technology was proposed. In 2019, the correction of point mutation by prime editing